All right. We're just got started on the sulfur cycle, and it's a very complex cycle, but it's important because it's part of what we build our bodies with. It's also an important chemoautotropic processes associated with it. And one of the really important things is that sulfur can either oxidize abiotically or biotically. And this is why acid rain occurs, that if there's combustion of sulfur-containing compounds, organic sulfur-containing compounds from coal or other fossil fuels, then that sulfur goes into the atmosphere as sulfide, and then it's converted into sulfate in the rain, and sulfate is actually going to be in the form of sulfuric acid. So it causes acidic problems. And we'll get to that when we talk about toxins. But also bacteria can do it biologically. So there's this whole series of sulfur transformations, dissimilatory sulfur reduction, and this is essentially the equivalent of denitrification, where it's an oxidation of organic carbon with sulfur, rather than starting with sulfate and rather than nitrate or oxygen for respiration. There's also an interesting one in that thiosulfate has sulfur that's at two different redox oxidation potentials, and in splitting it, a microbe can gain energy. So there's a very interesting chemoautotropic. In addition, sulfur precipitates, hydrogen sulfide precipitates with metal sulfides to create deposits. And then finally, there's anoxygenic photosynthesis. So we talked about this already. There's an ancient form of photosynthesis that was around before organisms could split water and create oxygen. They use sulfide as electron donor, and it produces sulfate. So here is, I'll go through all these again. And it's like I said, it's complex. One of the things I think that's really interesting about this is this shows how evolution has led to essentially using every different type of compound in these elemental cycles that's involved in biology. So organisms have adapted to oxidize each step of sulfide, from sulfide to sulfur to thiosulfate to sulfate, in oxygen containing environments. And this is going with or against potential energy. Good, Sean. It's going with potential energy, right. It's going from left to right in the oxic environment. And then they've also evolved ways to use all this stuff and use it to oxidize organic carbon in anoxic environments. This anoxygenic photosynthesis is actually going against potential energy, right? So that takes light to push it. So that's how it can be going the opposite way. The disproportionation is going with potential energy because it is moving, it's producing an oxidized sulfate, but it's also producing even more reduced sulfide. So this is basically a bigger gain than this is in terms of energy shifts. Dimethyl sulfide is globally important, and it is a byproduct of a toxin that phytoplankton put in their cells. And if a phytoplankton cell is eaten, it creates this toxin that lets go of sulfur gas. And so there's actually this two methyls and a sulfide on it is a gas, and it goes into the atmosphere, and it also eventually becomes oxidized either biologically or abiologically. But it's a globally important thing. It's more important globally because of marine phytoplankton than freshwater, just because there's so much more of it. And then there's another really interesting thing that is going to link the sulfur cycle to the metal cycle, and then we'll link the metal iron cycle to the phosphorus cycle. So it's going to be an important linkage across all the different cycles. Does anybody know what the common name of iron sulfide is offhand? It's also called pyrite. And then the common name of that is, you may know what iron pyrite is, the common name of iron pyrite. Got it, Jaden got it, great. Fool's gold, right? So maybe you've seen fool's gold before. It looks sort of gold, but it's not soft like real gold is. And yeah, so this stuff precipitates in anoxic sediments. When it's in a fine particulate form, it looks black. And so that's why anoxic sediments, when you pull them up, they do two things. One, they smell of sulfide, they smell of rotten eggs. And two, they have that black coloration, and that's just little particles of iron sulfide that are in that. And so that's how you can see if the sediment's really reduced or if it's oxidized. In addition, their fermentation is also called putrefaction, and the smell of sulfide, somewhat organic sulfur compounds, but also sulfide, is one that we're extraordinarily sensitive to. And that's basically because in anoxic, when bacteria operate on proteins, they release sulfide. Why would humans have evolved an aversion to that odor? Anybody have a guess on that? To avoid bacteria. Yeah, sulfide is poisonous at high concentrations, but we can detect it at just a molecule or two in a liter of air. I mean, it's actually the compound I think we're the most sensitive to. But Jaden got really pretty close to the right answer. If something is rotting meat, right, rotting proteins, then there's bacteria and the food might be contaminated. And so that's why rotten meat smells so bad. That's why cadavers smell bad. That's why rotten fish, part of the reason that rotten fishes smell so bad. And the ammonium and the sulfide are two of the things that really turn most of us off. The next cycle is much simpler in contrast, phosphorus, and that's because there's fewer forms of phosphorus that can be at different redox states. So there's organic phosphorus, and then there's phosphate. The phosphate cannot probably not be used to oxidize organic carbon by most organisms, and because it ends up as phosphine, which is a toxic compound. And so organisms that did that, like they use nitrate, right, or sulfate to oxidize organic carbon, but they don't tend to use phosphate. So this is a lot simpler because organisms can just take it up as phosphate. They can mineralize it, and there's an enzyme called a phosphatase that just takes organic phosphorus and converts it into phosphate. And a really important one, though, is it precipitates with oxidized iron. And we'll get to that as we talk about the iron. Okay, so this is going to come back to us when we talk about eutrophication, and it's an important interaction. On the top, we have a lake that's anoxic all the way up to the top of the hypolimnion, to the metalimnion. In the bottom, we have a lake that's anoxic in the sediment, but it is at the very top of the sediment, and the oxic is oxic as well as the entire water column. Which of these is the eutrophic lake, and which of these is the oligotrophic lake? So which of the two lakes, A or B, is oligotrophic? This could be a question of the day. You might as well all type it in. Which of them is more oligotrophic? Did I say eutrophic or oligotrophic? Oh, okay, eutrophic. Which one's more eutrophic? That's why everyone's saying A. Good. Right. So we've talked about this several times now when talking about fish kills, talking about iron going into the building up in anoxic conditions, and we've already talked a little bit about iron precipitating out with oxygen, ferric hydroxide, but also iron precipitates with phosphorus. And this becomes important because if we have an anoxic hypolimnion, the iron that precipitates with phosphorus settles down and it then dissociates into iron and phosphate. And so in a lake that is stratified, when it mixes, all the phosphate comes back up again. And so if the lake is limited by phosphorus, then if the algae in the lake are limited by phosphorus, then this will stimulate their growth. Otherwise, when phosphorus sinks out in an oligotrophic lake, and once it gets down in the sediment, right, it will dissociate, but as it diffuses up to the top of the sediment, when it hits the top of that anoxic zone, this oxidized microzone, you can call it, it reprecipitates. So it's never able to diffuse out into the water column. So when this lake turns over in the fall, it's not going to push a bunch of phosphate back up into the water column. It's called internal loading. We'll talk about it again when we talk about eutrophication, but this interaction with iron is extremely important because it locks the phosphate into the sediment as well as the iron. But iron's much less likely to be limiting primary production than is phosphorus, which again we'll talk about in a few lectures. Okay, we have other trace nutrient cycles. We'll talk about silicon, and we've talked about it being in diatom frustules and in sponge spicules, as we have in the image behind me, and it can become limiting in lakes. And then we've talked about iron several times already in the ferrous iron. Note that the iron cycle in the book is not correct, and the labels ferric and ferrous don't match the actual, they're flipped. They should be in the other place. Organisms can oxidize iron, and it's an important chemoautotrophic pathway. And so it has to happen at oxic and anoxic interface. Have any of you gone over to the Tuttle ponds on the side of the dam? There's some little streams in there, and has anybody noticed what color those streams are? Okay. Yeah, tan. They're actually kind of orangish, right? Tan or reddish orange color. And so, yeah, what it turns out is going on there is that those streams are drains to drain the moisture out of the dam, and it's anoxic down in that groundwater that's in the dam. As it comes out, it oxidizes. The microbes oxidize it. So that's actually ferric hydroxide rust that the bacteria have converted the reduced iron and combined oxygen and created that ferric hydroxide. So that's a really thick mat of bacteria that you're seeing there. So if you ever get up there, it's sort of where the campground is, kind of along the edge of the dam, and have a look at that. You'll also see groundwater seeps and this orangish stuff like coming into rivers and stuff like that are quite orange, and then we'll see it with the hot springs as well. So we know that oxidized iron precipitates with phosphate and dissociates again in anoxic conditions. And the last sort of important issue is that chelators keep iron in anoxic solutions. So it turns out that if you, for example, take an iron supplement, it has EDTA in it usually as a vitamin. And the reason for that is it's chelating it and it's keeping it from turning into ferric hydroxide. And as it goes, as you eat it. These organic compounds can keep enough iron in solution to keep phytoplankton from running out of iron. If there's too much chelator, iron can actually become limiting, and this is the case in very humic, like blackwater swamps and such, where there's just so much organic carbon that the iron associates with it that can't get into the algal cells. Okay, then this is a little slice of a silicon graph, which is actually quite long, and the scientists at Lake Windermere figured out many of these nutrient cycles, the iron cycles, really pretty early on in the 50s, which is pretty amazing. That whole sulfur cycle was figured out in the 50s, and it's pretty early for biogeochemists to have figured out such a complex cycle. But the other thing they did is they measured everything. So they measured a lot of elements. They measured silica for years. And there's, I think, a 20 or 30 year series of this at least, but I've just chopped a few years out of that. And that's in the top. And then the bottom is Asteronella, which is a planktonic diatom. And what we can see happening here is that every spring, the Asteronella blooms. The populations go crazy after the winter, and it's usually in spring when it's spring mixing before stratification occurs. And when that happens, the silica in the water column, the concentration drops drastically. You can see it it sucks it down. Then, as that bloom goes away, and we'll talk about this seasonal succession further on in the class, the cells start sinking, and they break down and release their silicate into the water. And so we see throughout the winter, it builds up as they release the silicate. And then the next spring, diatom bloom sucks it back down again. And we get this nice up and down cycling of silicate. This can be important. One of the worries about eutrophication is that it stimulates diatom growth, and so the silicate doesn't work its way down through the large rivers of the world and into the ocean, which shifts the marine communities away from diatoms. And diatoms are really good food for the plankton, for the zooplankton. And so it basically shifts communities to being more being more recalcitrant to grazing and away from being as productive. So here's a concentration of silica at the depth of time in a hypereutrophic wintergreen lake and an oligotrophic lake, A and B. And we see that in the winter time, the concentrations are even throughout, even though it's somewhat stratified here. And we see the silicate building up in the stratified lake, the bottom of this stratified lake. Because the diatoms are blooming here and sucking the silicon down, and then it mixes back in, right? So we get it building up at the bottom of this lake, and it coming out of the sediments. In contrast, in the oligotrophic lake, it doesn't tend to build up as much until later in the season, because the diatom blooms stay in the water for a lot longer period of time. So we have the really high concentrations here, and then as it mixes, it mixes back in and the diatoms take off, and they start drawing it way down in here.
So did they model this with like the same stuff that they used for like the snowshoe hare and the lynx, like with like the spikes and stuff, or is this all like biochemistry when they modeled the diatoms?
Oh, so all that is just straight measurement. So you could draw an analogy with the hare and the lynx. Basically, they got those hare and lynx, but they went out and measured populations of hares and populations of lynxes, right? In this case, they measured how much silicate there was and how many diatoms there were, and saw that seesaw. It's driven by the physics of limnology, though, and light, rather than food availability for the hares and the lynxes. Okay, so I sort of promised you that I'd talk a little bit about the linkages among these near the end and come back to it. And this is a really interesting case study here, and I always talk about it in class, and I often ask people to about it in exams or quizzes because it illustrates that understanding the biogeochemical cycles can have real implications for the real world. And this is a story from a peaty lowland wetlands that are in the Netherlands. And what they wanted to do is go back to the native condition of high macrophytes and no cyanobacterial blooms. And so there was a eutrophication problem, phosphorus pollution that got in there. And so what they decided to do was put in water that they treated to reduce the phosphorus from the river. But when they did that, the water also had high levels of sulfate in it, which they didn't realize. So they spent a lot of money treating this water that they put into the wetland. As the water got into the wetland, the sulfate got reduced to sulfide in the anoxic sediments, which then precipitated the iron. And it also, all the iron, it also poisoned the roots of the macrophytes. The sulfide itself is toxic. Once the iron was locked up, and what did it precipitate the iron as? What form is what compound is that? Anybody remember? The compound actually. It's ferrous, FeS, right? Good. So fool's gold, pyrite. It precipitated the iron out. So the iron wasn't present to bind the phosphates, and the phosphate concentrations increased, even though they're putting low phosphate water in. So we can go back up here and link these together. So basically, when they got rid of the iron, right, the phosphate could come up here, but it wasn't going to precipitate anymore with the iron. So it was able to go back into the water column and stimulate the algal blooms. And we can look at the metal, the sulfur cycle, why that links to the sulfur cycle. They put a lot of sulfate in the anoxic conditions. It was used to oxidize organic carbon, converted to sulfide. Some of that sulfide was toxic to the macrophytes, and some of it precipitated out the iron, right? So now we've linked the sulfur cycle to the iron cycle to the phosphorus cycle and how the whole system works. Had they understood this, they wouldn't have spent all the money treating the water for phosphorus, or they would have figured out a way to get the sulfate out before they put that water in, and it would have saved them a lot of money and grief. So the very last part of this lecture is trying to link all these things together to get you to really realize how all these things are related to each other. So if this could be meters or centimeters in a sediment. It could be a lake, anoxic or anoxic. And we see that we have but we'll just think about it maybe being a sediment that's anoxic at depth. And then as we move down, the redox drops as well. So we have oxygen here, high redox, no oxygen here, low redox, and the redox keeps going down. Here's what we just talked about, the iron phosphate being oxidized and stuck at the top, right? And the phosphate concentrations increasing down at depth and being low up here. We also just talked about the sulfate, and we have oxygen around. The sulfate's going to be high, and it's reduced, used to oxidize organic carbon to hydrogen sulfide, precipitating out the iron, making iron sulfate. So we have this oxidized microzone where we have sulfur oxidizers and sulfate reducers. The sulfide is diffusing up into the oxygen containing water, and the microbes that oxidize, chemoautotrophs that oxidize the sulfur, are able to do well. The sulfate is diffusing down into the sediment after it's created, right? And the organisms then can take that sulfate and reduce it to sulfide as they oxidize organic carbon. So we'll have this high microbial diversity at the oxic-anoxic interface. Similarly, we can have nitrogen. We have low ammonium in water. It's all converted to nitrate by nitrifiers, right? So the nitrate tends to be high, the ammonium low in the oxygen-containing environment. But as you go to the anoxic environment, ammonium has less potential energy, so everything ends up going into the form of ammonium. And this oxygen, oxic, anoxic interface, we have the denitrifiers, where the oxygen has to have disappeared, and they'll use the nitrate to oxidize organic carbon. Fe3+, ferric hydroxide precipitates as ferric hydroxide and or ferric phosphate. And then we have Fe2+. So we have reduced iron, oxidized iron. And when that reduced iron, diffuses into the environment, it can be combined with oxygen by iron oxidizing bacteria. And then at the very lowest redox, methanogens can, and the sulfate has to be gone. So you'll see the sulfates there, then the redox drops low enough that the methanogens can start taking CO2 and converting it to methane. And the methane concentrations are high down there. It diffuses out. As it diffuses out of the sediments, the methanotrophs will eat it up. Unless this happens to be a wetland where there are plant roots that are down, and then the methane will get into the vascular system of the plant and go up and out through the stomates and miss the methanotrophs. So this is gives you an idea of sort of the overlap and interactions of the nitrogen, the phosphorus, the sulfur, the iron cycles, and that there's a really high activity zone with it at this oxic and anoxic interface. And it's because of the potential energy that we talked about, right? Because when there's a reduced compound in an oxidized environment, it has potential energy, and the microbes are going to use it. When there's an oxidized compound in a reducing environment, it has potential energy, and the organisms will use it. If these two are close together in a sediment, right, you can get the diffusion of the reduced into the oxidized, and you can get a diffusion of the oxidized into the reduced. So it basically is putting fuel in for these organisms to operate. And people have actually taken advantage of this and can make microbial batteries out of it. And there's some cool things like cable bacteria that are long enough that they can actually move electrons back and forth between the two. And we can maybe talk about that some as we move into unusual habitats. So this ends this chapter.